Hi, hello, good morning everyone. I'm Dr. Harika Kishore. On behalf of Vista's Learning, I'm here as your biology educator. Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the live classes, recorded sessions, concept videos, animated videos. Moreover, you're going to get this lecture notes in the form of PDFs. Get your acquaintance with all the channels available and make sure to subscribe to the channel Vista's Learning Class 11 and 12. If you find this session useful and interesting, click on the like as well as share this session with your friends. And also click on the bell icon such that you are going to get all the upcoming video notifications. Saying that, let's begin our today's session. We are in the series of the sessions where we have been discussing about the chapter that is molecular basis of inheritance, which comes under the unit genetics and evolution, right? In the last session, we discussed about replication, how it is going to be happen by means of semi-conservative mode of DNA replication. After that, let's move to understand right? Now, this is the outlook of molecular biology of central dogma, where DNA, whatever the genetic information that is stored in the DNA, is going to transcribe into RNA. As this RNA is useful to transmission of the genetic information which is coming from the DNA, it is termed as a messenger RNA. This genetic information which is present in the DNA is first going to be transcribed into mRNA, and this process is called as transcription. So in the last class, how the DNA is going to replicate to make its own copy, that we discussed under the topic process called as replication. Once the replication is done, now the DNA, whatever the genetic information which is stored in the DNA, is now going to be transcribed into RNA. And that process and that copying of the genetic information which is present in the DNA into RNA, we call it as a transcription. And this is a second process what we see in the molecular basis of central dogma. Then, now the information, genetic information is present in the mRNA. By making use of this genetic information, now the proteins are synthesized. Synthesis of the protein takes place by using this information. The sequence of the amino acids are arranged to produce a particular protein, and that process, what we call it as a translation. Now, we are at this process. What is that? Transcription. So, the process of copying genetic information. We well know that the genetic information is stored in DNA. Now, this is the process of copying. Copying down of what? Copying the genetic information which is present in the, which is stored in the DNA from one strand of the DNA. Here, the genetic information will get transcribed from one strand of the DNA into RNA, and this is termed as, and this process is termed as transcription. Here the point is, when the replication is taking place, replication takes place toward the length by the forming a small opening, and that opening is called a replication fork. By forming the replication fork, there is an unwinding this coiling of the DNA helix opens up to form a small opening, and that is called as replication fork. And that is the site where the replication happens, and this replication of the DNA extends towards the entire length of the DNA. But that doesn't the same case when you consider the process of transcription. What is the change that is coming from replication to transcription? Here also, there, while we are dealing about the DNA replication, that is also just because of like base pairing, what we call it as a complementary base pairing. Here also, the principle of complementarity, governs the process of transcription. Just like the complementary base pairing. What do you mean by complementary base pairing? We already know that purines pairs up with pyrimidines and pyrimidine pairs up with purines. For example, adenine, it is a purine which pairs with the pyrimidine. But here the thing is, the pyrimidine thymine is going to replace with the uracil, because uracil will be present in the RNA and thymine is present in the DNA. So here we already know that thymine present only in DNA. Therefore, adenine pairs with uracil, which is present only in RNA. Right? So here also the principle of complementarity grounds, that means it will helpful for the complementarity, that means the polymerization of the nucleotides that are going to be present in the mRNA. Except, just now we mentioned that adenosine complements, instead of complementing with the thymine, it is going to complement with the uracil. That happens in the case of RNA. Here, adenosine complements, now forms base pair with uracil instead of thymine. Instead of thymine, here in the RNA, that is an mRNA, we see the presence of uracil. That is the difference. Next, moving forward, here, just like we said, while we are discussing about the DNA replication, right. This is how the formation of the daughter DNA takes place, by taking the parental strand as a template strand. This is a template strand. Right? And this is a newly formed strand. This is a newly formed strand. New strand. So here, however, unlike in the process of replication, which once started off, polarity takes place and the formation of the new strands takes place using the parental strand as a template strand, and this takes place throughout the length of the DNA, and that too, both the strands will act as a template. Here, the total DNA of the organism get duplicated in such a way that the total length of the DNA, total DNA of the organism is going to be produce its own copy, replicate, that is duplicate. In transcription, only a segment of DNA, not the entire length of DNA, that is going to be transcribed into mRNA. Instead, see, if you consider this is the entire length of DNA, okay, this is the entire length of DNA, for example. Not the entire length of the DNA that is going to be transcribed. Then what happened? Only a particular segment. Consider this is a particular segment. Okay, only a particular segment, and that too only single strand. Out of this, only single strand it is going to be transcribed into mRNA. Here the point is not the entire length of the DNA, not the two strands of the segment of the DNA it is going to be translated, but only one strand in the segment of the DNA that is going to be transcribed into mRNA. This is completely unlike to the case of DNA replication. So this point denotes like, however, unlike in the process of replication, which once set in, the total DNA of the organism get duplicated, that we seen in the case of DNA replication. But here in transcription, only a segment of DNA. This is a segment of DNA. In that too, only one of the strand, only one strand is going to be copied, is going to be transcribed into mRNA. RNA. Why we are calling it as an mRNA? Because it is using as a messenger. That is what mRNA. Now, so this necessitates we need to decide the boundaries. We need to know the boundaries from which point to which point that is going to act as a segment of the DNA. So here this necessitates defining. We need to define the boundaries that would demarcate. This is the boundaries in this example. This is the boundaries and that demarcate the region. This is the region that is going to be copied, that is going to be transcribed. And the strand of the DNA. Out of two strands, which strand is going to be transcribed? That is also have to be decided. So, and the strand of DNA that would be transcribed. So, as the entire length of the DNA is not going to be transcribed, it is only the segment of the DNA. In that also, not the complete two strands of the segment of DNA, but instead only one strand of the segment of the DNA that is going to be transcribed. So, it is necessary for us to first define the boundaries and also to decide which strand of the DNA that is going to be transcribed. Now, moving ahead, here is a question. Just now we said that in the segment, this is a small segment, not the two strands, this is one strand and this is another strand, right? Now the two strands are going to be transcribed into mRNA. If that happens, here we'll get one mRNA and the other strand of the DNA also acts as the template to produce another mRNA. So here we are going to get two single strands of mRNA which are complementary to each other and they are going to form a double-stranded mRNA which we don't need at all because it cannot translate and thereby there is no production of the protein takes place. So here the reason is, answer for this question is very, very simple. Why both the strands are not copied during transcription of DNA into mRNA? That has a very, very simple answer. To understand this, let's take a simple example. So here is a sequence, double-stranded sequence of DNA. So consider this is a DNA. Okay, DNA. Now, we separated the two strands. Here, we are about to understand why the both strands of the DNA are not transcribed into mRNA. If that happens, what it is going to be resulted in? Let's understand that. Here is one strand, right? And here is another strand, right? If it transcribes into mRNA, here we'll get mRNA. And this is the second strand of the DNA. Again, it is going to transcribe into another mRNA. Let's take, so here as we already know that if adenine is present in DNA sequence, in the mRNA sequence, it is going to be uracil. G, G, A, G, U, C, C and here that is U. Now, so this is the sequence, right? Now, right? So this is the first mRNA what we got where the first strand of the DNA acts as a template. Same way, the second strand of the DNA, if it is again going to act as a template, see, this is the same strand. TGGAGT, TGGAGT, CCT, CCT. Then if you take the nucleotides which comes in the mRNA like A, C, C, U, C, A, G, G and A, right? This is a sequence. Now, here is one mRNA, right? Okay. And here is an other mRNA, right? Let's look at the sequence of the amino acids, what this mRNA is going to translate. So we know that 3 nucleotides combine together to form one codon. So what is the first codon we have? That is UGG. Let's look at in the genetic code chart that what is an amino acid that codes from a UGG, right? First letter is U. So here is the first letter U. Second letter is G. Here is the second letter G. So here. And what is the third letter? Again G. So G will come here, right? We have UGG. UGG codes for tryptophan, right? Same way, another second codon in this sequence, that is AGU. First letter A, second letter G, and third letter is U. U codes for serine. Similarly, CCU, first letter C, second letter C, third letter U, proline. So this is a sequence of amino acids that are coded by the first mRNA. Let's consider this is the first mRNA and this is the second mRNA. Okay, what is the sequence of amino acids that are translated by the first mRNA sequence? That is tryptophan, serine, proline. Tryptophan, serine, proline. Similarly, we'll check for the amino acids. Okay. So here ACC, right? ACC. First letter A, second letter C, and third letter is again C. ACC. Threonine. Threonine here. Next, UCA. First letter U, second letter C, third letter A. U, C, right? A. Serine. Done. Last codon. GGA. First letter G. Second letter is also G. Third letter A. What is that? Glycine. What is the sequence of the amino acids that are coming by the mRNA2? That is threonine, serine, glycine. So here is the difference. The first amino acid sequence is tryptophan, serine, proline. Second one is threonine, serine, glycine. So there is a difference in the sequence of the amino acids that are coded by the mRNA that comes from a single segment of DNA. And if you look at the complementarity that is present in the two strands, that is the two mRNA strands that are transcribed by a single segment. Here is the first sequence mRNA and this is the second single-stranded mRNA. If you look at these two strands, these are complementary to each other and there is a chance of forming two double-stranded. Look at here what happened. RNA, double-stranded RNA because it is having a complementary base pairing with each other. As the two strands are complementary to each other, they form a double-stranded mRNA. So if the double-stranded mRNA is produced, it is not going to translate into protein. No translation occurs, no protein produced, because the two single-stranded mRNA combines, as they are having a complementary base pairing between them. They base pairs by forming hydrogen bonds in between, 3 hydrogen bonds, 2, 3, 2, 3, 3, 2, and this is a double-stranded mRNA where it doesn't further translate and it doesn't produce the final protein as a product. So we understand this process. Now, after seeing the example, it is very easy to understand the statement here. The question is why the both strands are not transcribed into mRNA, why both the strands are not copied during transcription, has a simple answer. Here is the first reason for that. If both strands act as a template, if both strands are going to act as a template, they would code for RNA molecule with different sequences. That means, as we already know that complementarity doesn't mean the identity, that means identical single strands, because here we have a uracil that is going to be present in the RNA, and there the thymine will be present in the DNA. And in turn, if they code for proteins also, the sequence of the amino acids in such proteins would be different. That is what the sequence here we have different sequences. Now, second one is, if both the strands of the DNA are going to transcribe into mRNA, they are going to produce different sequences of the amino acids that are going to present in two different proteins. So hence from one segment itself, consider this is a single segment. Hope it is visible to you. This is a segment of DNA, right? Where both the strands are going to transcribe into mRNA. Now this will produce one protein. Right, today it is giving. And second, it is going to produce another protein, say for example two different proteins, protein 1 and protein 2. And this would complicate the genetic information transfer machinery. Next, second reason, the two RNA molecules, if produced simultaneously, definitely would be having complementary to each other, hence would form a double-stranded RNA. That is what we have shown in the image and the last slide. If the double-stranded RNA is going to be produced, there is no protein synthesis. If the protein is not going to synthesize, then the entire process is going to be not useful. So here, this would prevent the RNA from being translated into protein, and the exercise of the transcription would become futile, which is not useful because it is not ending up with a useful protein synthesis, but instead there is no protein synthesis that is going to be happened. So with this we have understood the concept why both the strands of the DNA are not acting as a template and why both the strands are not going to be transcribed into mRNA. Instead, only a segment of DNA, in that also only one strand is going to transcribe into mRNA. Now, moving on to transcription unit. Now, so if you consider this is a segment of DNA, in the segment of DNA we have three regions. Three sites. So here this is considered as a transcription unit. The transcription unit in DNA is defined primarily by the three regions in the DNA sequence. This is a segment of the DNA where the first one is called as promoter and the second one is a structural gene, third one is terminator. And promoter is a site where the transcription initiates and terminator is a site where the transcription ends. So here promoter, structural gene and terminator. Let's understand more about this promoter, structural gene and terminator. So there is a convention in defining the two strands of DNA in the structural gene of a transcription unit. So just now we said that this is a segment of the DNA where we have promoter, structural gene and terminator. While we are discussing about the transcription, that means replication of the DNA, we mentioned that the direction of the polymerization is always 5' to 3'. Here also, same polarity is useful for the RNA polymerization also. So here, since the two strands have an opposite polarity, the DNA-dependent RNA polymerases, there the enzyme is DNA-dependent DNA polymerases because the synthesizing strand is also DNA and the template strand is also DNA. So DNA-dependent DNA polymerases. But here as DNA is depending on here. In the previous case, DNA is depending upon the DNA. Here, the produced one is mRNA and RNA is depending on DNA. One of its strand is going to act as a template. So the enzyme will be definitely DNA dependent. Synthesized one is RNA. So RNA polymerases. DNA dependent RNA polymerases also catalyze the polymerization in only one direction, and that is, this is very, very important. Here also the direction of the polymerization is 5' to 3'. This is very, very important to remember. Now, as the polymerization happens from 5' to 3', then the template polarity should be 3' to 5'. The strand that has the polarity 3' to 5' acts as a template and it is referred to as a template strand. It is going to refer as a template strand. So look at here. One strand will be running from 3' to 5' and the opposite will be running from 5' to 3'. So here the other strand, say for example, this is running from 5' to 3'. This is running from 3' to 5'. As it is having the opposite polarity to the direction of the polymerization, that is 5' to 3', this strand is going to act as a template and that is what we call it as a template strand. Coming to the next strand, another strand, the other strand which has a polarity 5' to 3', 5' to 3', and the sequence same as the RNA which is produced, except at the position of thymine it will have uracil. This strand is going to be displaced during transcription. Out of two strands, only one strand is acting as a template, right? Now, strangely, this strand, which is not coding for any mRNA, called as a coding strand, which doesn't code for anything, it is referred to as a coding strand. I'll explain why. So here, we mentioned that the strand which is not coding anything, we call it as a coding strand. But the strand which is coding for mRNA, we call it as a template strand instead of coding strand. Why it is happening? The reason here is, see, all the reference point is with relate to the coding strand. This is another important point to be noted. All the reference point while defining the transcription unit is made with reference to the, we have to see in accordance with the coding strand. And here in this example, this is coding strand. See, here we said that 5' to 3', this is the direction of the polymerization. and the strand which has an opposite polarity, that is 3' to 5'. Out of these two strands which is having 3' to 5', this strand 3', 5'. So it is going to act as this strand is going to act as a template strand. But the other strand which has a similar polarity, that is 5' to 3', 5' to 3', we call it as a coding strand. But particularly here in this case, this strand is not going to code for anything. But why we call it as a coding strand? Because the reason here is, if we take this strand as a template strand, we'll get mRNA, right? We'll get mRNA. So here we have U, A, C, G. Here, in the place of T, as it is mRNA, we'll get uracil. And A, C, G, U, A, C, G, U, A, C, G, right? U, A, C, G, C, G. See, if you look at the sequence, this is similar to the sequence of the coding strand. Except at the place of thymine, we have uracil. At the place of thymine, you have uracil. At the place of thymine, you have uracil. Instead of this difference, the entire sequence is similar to this sequence, AC, ACG. And at the place, you have uracil, and again, ACGU. Here, the same thing, ACG. You know, because here it is T, thymine. Instead of that difference, the entire sequence of the coding strand is similar to the mRNA. Now, this is the strand. This is mRNA, which is further going to translate, show translation, yes, to protein. But not this strand, it is going to convert into protein. But instead, the sequence which is present on the coding strand, which is very similar to the formed mRNA, that is going to produce protein. So that is what we consider this strand as a coding strand, because the sequence which is present on the coding strand is ultimately going to decide the sequence of the amino acids that are going to appear in the protein. So now by this we know that though the coding strand is not going to literally code for anything, but we call it as a coding strand because it is having a similar sequence to that of the mRNA that are transcribed by template strand. That is it. So here is a hypothetical sequence which is given to you to make you understand what is the polarity of the template strand and what is the polarity of the coding strand, right? Now, moving ahead. So, here is a representation, we already discussed about it. Here, we have on either side of the structural gene, we have a promoter and we have the terminator. The defining of the promoter and the defining of the terminator is all with reference to the coding strand. This is a coding strand. Okay, let's move ahead. So here, the promoter and terminator, we have promoter, we have terminator, flag that is present on either sides. Either side of what? Either side of structural gene. So here is the structural gene. On either side of the structural gene, we have promoter one side and terminator another side in the transcription unit. Now, the promoter is said to be located towards 5' because the reference is towards coding strand but not the template strand. Wherever the 5' end is present on the coding strand, towards the 5' end, that we call it as an upstream, downstream. Towards the upstream, see, the promoter is said to be located towards 5' and this is upstream. Upstream of the structural gene. This is the structural gene. Now, the reference is made with respect to the polarity of the coding strand. This point I already mentioned. Reference, always our reference is made with respect to the polarity that is present on the coding strand. On the coding strand, towards the 5' end, we have promoter, and towards the 3' end, downstream, we have the terminator. Now, Promoter it is a DNA sequence. It is a DNA sequence. Nothing but the promoter is a DNA sequence that provides a binding site for the enzyme. What is the enzyme? DNA dependent RNA polymerases. RNA polymerases. This is an enzyme, and it is the presence of the promoter in a transcription unit. This is a transcription unit. We have the promoter here, and we have the terminator towards the 3' end of the coding strand. Right. Now, that also defines the template and coding strand. Here, by switching its position with terminator, by switching its means promoter, where we have promoter here, where we have terminator here. What they are mentioning, by switching, by swiping, by exchanging, take the promoter here and take the terminator here and send the promoter there. Switching, we have swiped the positions of terminator and promoter. By now, we got the terminator here and the promoter there. What we said, coding strand 5' is a position of promoter. Now, the promoter went to this side. So, the coding strand 5' will come here and 3' will come here. That means, see, by switching its position, it means promoter. By switching the promoter position with the terminator, we did it already, the definition of coding and template strands would be reversed, because the 5' of the coding strand comes towards this side because the promoter here. So this will become coding strand and this will become terminator. See, I'll explain again. This point is important to understand, but it is easy. We switched the position. We have taken the terminator here and we send the promoter here. By switching its position, by switching the promoter position with the terminator, the definitions of coding and template strand would be reversed. Now, if the promoter goes here, this is the 5' and this will be definitely coding strand. Now, this is going to be coding strand. And this will be, because it has 3'. Template strand. So if you switch the position of the promoter with the terminator, there will be a switching over to the strands also. That is the point. Simple. Then the definition of the coding and the template strands would be reversed. That means whatever the coding strand now become template strand and whatever the template strand now become coding strand. If you exchange the position of the promoter and terminator, that is it. Now, so here, coming to the terminator, we know that the promoter will be present to the 5' end of the coding strand and that to upstream, and the terminator will be present towards the downstream of 3' end of the coding strand. The terminator is located towards the 3' end, downstream of the coding strand, because the reference is with respect to the coding strand, no? Now, and is usually defined the end of the process of transcription. We already mentioned that the promoter is a starting point for the transcription, and terminator, in the name itself it is there, which terminates, which ends up the process of transcription. Now, along with the promoter, structural gene and the terminator, we also have a regulatory sequences, which is also can be called as a regulatory gene. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Upstream or downstream to the promoter, and that sequence, what we call it as a regulatory sequence, and the details regarding to this regulatory sequence can be studied while we are dealing with the regulation of the gene expression. Some of the properties of the sequence, of these sequences, regulatory sequences, shall be discussed while we are dealing the last topic, that is the regulation of the gene expression. Now, let's move forward towards understanding the transcription unit and the gene. See, we know that gene will be present on the DNA sequence. That is obvious. There is no doubt about it. Genes will be present in the sequence of the DNA. Let's understand. A gene is defined as a structural, functional unit of inheritance, heredity, whatever the properties that are coming from the one generation, that is parent generation, to its progeny generation, that is daughter generation. So here a gene is defined as a functional unit of inheritance. This point is known and it is important. Move ahead. Though there is no ambiguity, there is no doubt at all that the genes are located on the DNA, still it is difficult to literally define a gene in terms of the DNA sequence. Why? Because see, any DNA segment, this is a DNA sequence which codes for either tRNA or rRNA. We call this as a gene. That DNA sequence as a gene. A DNA sequence coding for tRNA or rRNA molecule also defined as a gene. However, by defining cistron as a segment of the DNA, if you consider this segment of the DNA as a cistron, which codes for the polypeptides, see, coding for polypeptide, the structural gene in a transcription unit could be called as monocistronic or polycistronic. And we know that monocistronic will be mostly present in the eukaryotic, most advanced organization, that is eukaryotic organisms, whereas the polycistronic will be present mostly in the bacteria, that is lower organization, prokaryotes. So polycistronic can be present is seen in the case of prokaryotes, whereas the monocistronic will be present in the eukaryotes. So here in eukaryotes, the monocistronic structural genes have interrupted coding sequence. If this is a sequence, it will be interrupted in between by intervening sequences. That means it will show split gene arrangement. These genes are splitted by the intervening sequences. It will be having coding sequence, non-coding sequence, coding sequence, non-coding sequence. Here in the representation, if you consider this field as a exon, this exon is a coding sequence. We have again exon and exon. So 1, 2, 3. Okay. It is interrupted in between. This coding sequence is interrupted by the non-coding sequences, and this non-coding sequences is called as intron and intron. These introns are removed in the process of splicing, where it ends up with the formation of the mature RNA or processed mRNA. So now further, the exons, what we call it as a coding sequences, joins up. Only we can see the exons in the mature RNA or processed RNA. So look at here. In eukaryotes, the monocistronic structural genes have interrupted coding sequences. The coding sequences is interrupted by the non-coding sequences. The gene in the eukaryotes are split. That is what we call it as, there is a split gene arrangement. The coding sequence or the expressed sequence, we call it as an exons. So these are all exons. These exons are said to be those sequences that are going to be appear in the mature or processed RNA. This is the process of the splicing where the introns will removed off and the exons are joined up together to be finally appear in processed RNA or mature RNA. So this is called as mature or processed RNA. Now, so here the exons are interrupted. Just now I have shown how the introns are going to be. So if you consider this is an exon, this is an exon, this is an exon. These are introns. We have introns here. Okay. And these are all exons. Okay. So, the exons are interrupted. In between it is interrupted, no? Interrupted by introns. Introns are intervening sequences, do not appear in the mature or the processed RNA, because while the processing of the RNA, while the splicing process of the RNA is happening, these introns are cut and removed, whereas the exons are joined up together to be appear in the final processed or mature RNA. Now, this type of arrangement, we call it as a split gene arrangement, and the presence of this split gene arrangement further complicates the definition of the gene in terms of DNA segments. Though it is obvious, very clear, without any doubt, we can say that the genes are present on the sequence of the DNA, but further, the presence of this split gene arrangement makes it difficult to define the genes in terms of the sequence of DNA. Now, so here, inheritance of a character is also affected by the promoter and regulatory sequences of the structural gene. Hence, sometimes this regulatory sequence can also be defined as a regulatory gene. Regulatory sequences are loosely defined as regulatory genes. Regulatory sequences can also be called as a regulatory genes, even though these sequences do not code for any RNA or protein, right? Yeah, this is it for today's session, where we discussed about the transcription. In the coming up session, let's discuss about the type of the RNA and also process of transcription, right? Like, we're going to discuss about the different types of the RNAs like mRNA, rRNA, tRNA. Let's meet up in the next session. Until then, keep learning with We Learning. Thanks a lot.